Okay, thank you. I think you all know that tropical rainforests are the most diverse terrestrial ecosystem on the planet. They contain over 50% of species, and all this is packed into just 7% of land. Tropical rainforests are characterized by a forest which has a closed canopy, mainly growing along the equator, and one of the main characteristics is that it's dominated by angiosperms. And in rainforests, and I don't have to remind you here, palms are omnipresent and extremely important. Palms and rainforests go hand in hand. It's very hard when you go into a tropical rainforest of Southeast Asia or in South America to take a picture of the rainforest without including at least one species of palms. As you can see here in Brazil, in Southeast Asia, Indonesia, and Papua New Guinea, and even in Africa, where palms, as we've seen from the talk of Ingrid yesterday, are not really diverse. Palms are extremely omnipresent throughout the forest. And sometimes they can even come to dominate the understory. For example, here, as Adama showed, Podococcus barteri dominating rainforests in Gabon. But palms have also adapted to alternative biomes. For example, and here are just a few examples. Phoenix reclinata. I practiced before that. Phoenix reclinata growing in the highlands of the Cameroonian range, behind the Mount Cameroon at 2,000 meters elevation. Moisia flexuosa in the periodically inundated savannas of Bolivia. And the iconic Menemia, which grows in the Egyptian deserts in a very arid ecosystem. If we count the number of species that are restricted to tropical rainforests, that represents over 90%. So only a fraction of palms have actually been able to adapt to alternative ecosystems. I think we were all pleased as a community when this article came out, which estimated the size of populations of different species in the Amazon basin. And out of 20 most abundant species, 7 are actually palm species. And the most abundant tree species in the Amazon basin is the palm too. The relationship, the diversity of palms is highly correlated to water availability, and several studies have underlined this strong relationship. Here we have a latitudinal gradient diversity in the neotropics, and we see that as we go towards the equator, species diversity rises, and as we go further away, it diminishes. This relationship between diversity and precipitation is not just a recent relationship, it's also a past one. This very nice study in Malagasy palms has shown a very strong relationship between last glacial maximum precipitation and diversity of Malagasy palms, which is represented here, a very nice correlation. This relationship actually also extends over multi-million scale. For example, in Africa, a recent study has also shown that there's a very strong correlation between the Pliocene precipitation, so between 2.5 and 5 million years ago, and palm diversity. We can see that it increases as precipitation increases. We tried to understand the origins of rainforest biome, and we reconstructed the ancestral biome of palms, and we found that the ancestral biome of palms was significantly supported as being a rainforest. So here in blue, we have rainforest taxa, and in red we have alternative biomes such as mangroves, savannas, or desertic adapted palms. This relationship, or this result, is strongly based on the phylogenetic position of subfamily Calamoideae, which is here sister to all other palms, as we've seen in the talks of John and Bill. And most of the genera in this subfamily are restricted to the rainforests in Southeast Asia and in Africa. So one of the ideas that we tried to push forward was that palms might provide a very interesting and a very good model to understand rainforest diversification and rainforest evolution. First, as we've seen from the talks from yesterday and from John's introductory talk, We have an excellent knowledge of palm taxonomy, which is mirrored in the Genera Palmarum Volume 2. We have a worldwide checklist, and all this is available online in PalmWeb. Besides that, as I've shown, we have a very good idea of the intrinsic relationships between palms and rainforests. We have a very good fossil record, which is one of the best monocot fossil records amongst plants. And we also have a very good generic level phylogeny, which provides knowledge about relationships between genera and palms. So all this means that we have good data, and it puts palms in a pole position to understand rainforest evolution. One of the first questions we tried to answer with Bill was, well, if there's such a good relationship, what can palm evolution or origins tell us about the origins of this biome? For that, we used the super tree published by Baker and colleagues in 2009, and we mined the fossil record to provide a calibration point within these phylogenies. We selected 4 highly conserved fossils, which we then placed at different nodes, and we generated a dated phylogeny of palms at the generic level. And this provided an interesting result. The origin of all extant species, that means species that exist today, was dated to 100 million years ago, give or take a few million years. And this corresponds to the mid-Cretaceous. So palms diversified at the mid-Cretaceous, leading to a very nice long evolutionary history of about 100 million years. So how does this compare to the fossil record? Actually, it cooperates pretty well with what we know from the earliest occurrence of palm fossils or ambiguous fossils. And we have here for different types, for pollen, for fruit, for wood fossils, leaves, and also flowers. And we can see here in yellow, this is our estimated age of the origin of palms, and we can see that it corresponds pretty well to the occurrences of first fossils, and ambiguous fossils assigned to palms. In the second step, we try to understand the spatial origins of palms. Where did palms originate on Earth? And for that, we use different biogeographic methods. Now, these methods, when we're looking at a global palm phylogeny at a global scale, are not very precise, but they still provide an interesting result. Palms originated in North America and in Eurasia. And 100 million years ago, this is what the Earth looked like. And this was actually the Eurasian landmass, which was starting to break up. This also was corroborated by the oldest palm fossils, and we saw from Angela's talk yesterday, where she plotted the earliest palm fossils where they were mainly distributed in North America. So this is a nice corroboration of the biogeographic method with the fossil data. So we basically had 2 results. Palms originated in Eurasia 100 million years ago. And we used this result to suggest a hypothesis that rainforests started to assemble 100 million years ago in the north. If we go back to the fossil record, there's very little evidence of rainforests at 100 million years ago. But we did find one, which is a famous example of a paleoflora that has been described as having a rainforest-like vegetation. And this was the Dakota group fossil assemblage, which was found in North America, nicely fitting with our hypothesis of 100 million years ago and in Eurasia. Here I represent the number of families originating for millions of years, and this is based on an angiosperm-wide dated phylogeny, which was published by Susana Magalon and collaborators in 2015. And we can see here the origin of these different families. And 100 million years ago was actually a very interesting period in angiosperm diversification. This is also very well documented in the fossil record, where there was an extreme increase in different types of angiosperm families. Most of the angiosperm families originated at the mid-Cretaceous. So, as I said at the beginning, rainforests are dominated by angiosperms. So it might seem logical that the biome was started to assemble at a time when most of the families also originated. We then went a bit further and we estimated the dispersal events from Eurasia into South America and Southeast Asia. And we found our results suggested that there were several dispersal events into South America, into Africa, and all this happened before the end of the Cretaceous. Now, if you look at the evidence for tropical rainforest in South America, we also have a pretty good congruent pattern. The earliest fossil record that has been described as having a tropical rainforest-like vegetation was described here in Colombia, and it dates to 68 million years ago. So just before we're just at the end of the Cretaceous. And here we have an example of a fossil which is associated to piper, which is also which is a rainforest restricted genus throughout the tropics. Another more famous article that came out suggests that rainforests were already in place during the Paleogene, so after at the beginning of the Cenozoic, at about 58 million years, also from Colombia. Interestingly, these two paleofloras were characterized by low morphospecies diversity. So this is also congruent with the idea that rainforests were not full-blown rainforests as we know them today, packed with a lot of different species, but were just starting to assemble as a biome. We also have several indirect evidence to understand the origins of rainforests, and this comes from the molecular dating of other tropical rainforest lineages. The pioneering study of Davis et al. In 2005 dated the origin of Malpighiales, which mainly has families of restricted tropical rainforests occurring at, or most of them diversifying at 114 million years ago. And in another tropical rainforest restricted family, Ananaceae, which I work on a lot, we also dated the origin of this family to about 19 million years. So we're starting to get more and more data of tropical rainforest lineages which find their origins in during the Middle Cretaceous. There are also different types of vegetation simulation in the Cretaceous, and here we have one such study which suggests that tropical rainforests in the north is possible. So it's a bit hard to see, but here in dark green we have rainforests, but we also see that rainforests were possibly also growing in Eurasia at the time. So I think a few years on after we published this and thinking a lot with Bill and discussing these things, the main conclusion is that understanding the origins of rainforests is always going to be a difficult task. It's complicated, which seems self-evident. We have several ways of looking at rainforest evolution and origins. The first one comes directly from the fossil record, which is the only direct evidence that we can have of past vegetation types. But the fossil record is highly fragmented, both in space and in time. This is mainly linked to the fact that rainforests is a very difficult environment for fossilization to actually carry on. And then the other methods are molecular dating. Molecular dating allows us to understand the origins of biomes, which has been suggested by Toby Pennington and collaborators. It does provide some kind of information on the origins of these biomes, especially for those that are really restricted to them. But molecular dating is fraught with a lot of uncertainty linked to the estimation of diversification rates and substitution rates in DNA, and also to the uncertainty in the placement of the fossils that we have selected at different nodes. It also does not take into account the extinction. What we suggested in our article with Bill in 2013 is that we should use both approaches. They should be reciprocally illuminating to understand the origins of this complex biome. So in the second step, we were interested in understanding the tempo of diversification of tropical rainforests. How have species accumulated through time? For that, we extracted from our dated molecular phylogeny at the generic level the origin of major lineages through time. And this allows us to produce what we call the lineage through time, which depicts the origins of lineages through time. But of course, these methods are complicated to use, and in our case, we had a huge taxon undersampling. We only had one species per genus, so we underestimate diversity at a very high degree. However, we do have a very complete taxonomic sampling throughout all of the palm family, and so this suggests that we do have an accurate, or what we believe is an accurate picture of ancient accumulation of major lineages through time. So in order to prune our tree to only include the section of the LTT plot that we thought was accurate, we used different methods, and we came to an age of 25 million years. So after 25 million years, is not accurate enough to be used. We then use different maximum likelihood methods to fit different models of evolution, of diversification, and we found that the model that fitted the most this figure was a constant accumulation of major lineages through time. So we had a constant increase in diversification through time. Also notice that during the last mass extinction, there was no major or significant change in diversification through the KPG boundary. More recently, we used another method. I'm not going to go into the details, but it allows us to estimate speciation rates or diversification rates continuously through the palm family. And what we find, a very interesting pattern, is that our age of 24 million years or 25 million years corresponds actually to a period where we had an increase in speciation rates. So we have a model where we have a relatively constant rate, although slightly decreasing, throughout the early history of palm family, and then towards more recent time we have a huge increase in diversification. Alfred Wallace, we've heard about him already, suggested as an hypothesis that the large diversity of tropical rainforest could be the result of species accumulating, of a constant speciation through time with low extinction rates, mainly linked to the idea that rainforests were very old biomes and very stable ecologically. We know that that isn't really true. Rainforests are highly dynamic entities. And what we kind of suggested with Bill in our article is that there might be a mixed model of diversification. We have a background of ancient accumulation, of ancient constant accumulation of major lineages, and these lineages were able to survive even when we had major changes in climate because the rainforest was an ecosystem that never really disappeared. We've always had it. In contrast to northern ecosystems that have been majorly affected by glaciation, for example. And over this, we have pulses of recent diversification, which explains the actual present level of species diversity in palms. So this is a mixed model. And interestingly, similar patterns have been suggested in other tropical rainforests, restricted families, where we also have a background of major lineages accumulating with signals of recent bursts of speciation. And one of the recent ones that came out a few weeks ago was found in Mediaceae, which is a very nice example where we have this nice constant accumulation and then recent bursts of speciation, mainly corresponding to taxa that have diversified in tropical rainforests. And of course, there was the original article which suggested a mixed model of tropical rainforest evolution, which was found in leaf beetles. I just want to come back a minute to the KPG boundary. So if you remember, I showed you that there was no change in diversification, which was a surprising result because the KPG boundary is the last massive extinction event, and all plants or all organisms were thought to be affected by this event. So we did not find any evidence for that. And we didn't really discuss that because we estimated we didn't have enough data. But interestingly, other families have also been finding a similar relationship, especially those in tropical rainforests. Here, for example, in many Spermaceae, we have an increase in diversification rate exactly at the boundary of the KPG. And in Adnaceae again, we also found that there was no significant change in diversification rates. So recently, Silvestro and collaborators published a reanalysis, I would call it a reanalysis of the fossil data. So this has nothing to do with, at least from what I understood, nothing to do with molecular dating. They did a reanalysis of the fossil data using Bayesian methods, a very interesting analysis, and they also found a similar result where actually extinction rates throughout the KT boundary based on the fossil record did not change, whereas speciation and diversification actually increased. So this result shows that when we actually that the molecular dating results that we had at the beginning were showing towards some kind of interesting pattern that was occurring throughout flowering plants. So I was mainly talking about global diversification patterns in palms. I'm not going to go too much into detail about regional ones, but we heard from Jean-Christophe yesterday an extremely nice study which is starting to suggest that Astrocarium is becoming a very interesting model to understand rainforest diversification in the Amazon. And we heard that yesterday, suggesting that dispersal into different regions is a major driver in contrast to diversification by vicariance linked to geological models, geological uplift or dynamics. We also heard from Bill the studies of Christine Bacon on understanding diversification patterns in Southeast Asia, trying to explain the difference in diversity patterns throughout this incredibly rich region. And her works in triatrachycarpy have also suggested the very important role of dispersal into new regions which have triggered diversification, instead of maybe a vicariance by the appearance and disappearance of different island systems. Also, palms are becoming a very interesting model to understand the evolution of biodiversity in islands. And this was also something that Christine worked on very nicely during her PhD. So in the second part, in the second reflection, we was trying to understand the differences in regional diversity between major regions. Southeast Asia, Africa, and South America. I think we're all familiar with this pattern. Tropical America, huge amounts of diversity. Tropical Asia, huge amounts of diversity. And Africa, the poor odd man out with fewer species diversity. Palms actually represent this perfectly. We have high species diversity in South America, Central America, Southeast Asia, even Madagascar, and we only have to date 65 known species in tropical Africa. So there are many reasons to explain this disparity, and we've heard a few of them from Ingrid's talk yesterday about area. But one of the major ideas or major hypotheses over the past years is extinction. Africa has undergone very high extinction rates compared to other tropical regions. And to illustrate this in some way, in an article from Kissling and collaborators, they represented the evolution of rainforests extent throughout the Cenozoic. And you can see that for South America and in the Indo-Malay Peninsula, this hardly changed. It was a slight dip here. But Africa has undergone an extreme reduction in rainforest extent, especially accelerating from the Miocene onwards. This is also documented in the fossil record in Africa, where the studies by Pan and collaborators showed a decrease in morphospecies, especially throughout the Eocene-Oligocene boundary. So with our data in molecular phylogeny, we asked the question, do we find evidence for decreased diversification rates in tropical African palm lineages? If anything, a family such as palms should provide a strong signal of decreases in diversification rates. So we used a method at the time, which is called Medusa. And what Medusa basically does is that it goes through the tree and it tries to find nodes where an increase or decrease in speciation rates has to be implied to explain the actual species diversity. So it starts off with a constant diversification rate, and it identifies specific nodes where rates have to increase or decrease. And what we found is that we had 13 rate shifts throughout the palm family. Note first of all that most of these rate shifts are located younger or mostly younger than 25 million years. So again suggesting these rapid bursts of speciation in order to explain present-day species level diversity in rainforests. We found that there were no decreases in diversification rates. None of the African lineages showed any sign of decreased diversification rates. And all of the increased diversification rates were along Southeast Asian, South American, and even one Malagasy lineage. And I'll leave it up to you to guess which one that was. So this data suggests that it's more higher speciation rates in South America and in Southeast Asia, probably linked to the high dynamics of these regions, compared to Africa, that can explain present-day discrepancies between major regions in the tropics. So South America and Southeast Asia have higher increased diversification rates, and Africa just continues to diversify on a normal rate, mainly linked to the fact that Africa hasn't undergone major geological movements. It's been in a stable position for many millions of years, and where mountains tended to uplift, that always was in regions with high aridity. So it was linked to aridity. Again, looking at the Animaceae family, we also found a similar pattern. This was a study by Julien Massoni, which was a PhD student in Paris, and he looked at the diversification rate changes throughout the magnolias, so representing about 10,000 species. And when we zoom into Animaceae, we found that there was only a single rate increase to explain present-day species diversity, and that this rate increase was along a lineage which was mainly represented in Southeast Asia and in a few in the Neotropics. In an independent study, Bardin et al. From Jérôme Chave's lab in Toulouse looked at the evolution of Chrysobananaceae, which is another rainforest-restricted family with a very interesting pattern, also 80% of its diversity in the Neotropics. And they found, using a different approach than ours, that the hypothesis of higher speciation rates in the Neotropics was responsible for this pattern, instead of higher extinction rates in the Paleotropics. In a recent study from the Antonelli lab, they looked at global diversification patterns, and they interestingly found similar results to ours, where speciation rates in the Neotropics compared to Africa were higher. And it's not a relationship between extinction. So extinction in both cases was very high in the Neotropics compared to Africa. So tropical America once again gives a signal of higher speciation rates, and Africa doesn't show this higher extinction rate compared to other regions. I want to go back to the great Al Gentry, who actually suggested more than 30 years ago that rather than the flora of tropical Africa being impoverished, and I read this as having undergone higher extinction rates with respect to the Neotropics, the latter may be considered as uniquely and phenomenally enriched, to be read in my case as having undergone extremely higher speciation rates. One of the problems with our studies is that we're lacking information about what's happening at the species level. So we need species level phylogenies. So for that, with Adam Affe, we went around Africa collecting African rattans. African rattans are interesting because they form a monophyletic group, subtribe Ancestrophylaeni, and they have 21 species in there. So that represents 1/3 of all African palms. And so maybe this group, which is restricted to tropical rainforest mainly, might provide some interesting results. And so with Adama, during his PhD, part of his PhD and most of his masters, we generated a species level phylogeny. And I'm not going to go into all the details here, but what we found is that most of the species in African rattans are actually pretty young. So diversification really kicked off at about 10 million years ago. So we still have to apply different diversification methods to see if we do have a correlation or do have a link with extinction to explain these patterns. But it's interesting to say that even in the Neotropics, most species are young. So it really seems that most of the diversification, as I said from the beginning, is young, but that in the Neotropics there was some kind of boost, or in Southeast Asia some kind of boost, which led to higher species diversity, instead of relying only on extinction. Okay, now another interesting form of diversity in rainforest is growth form. For those who know rainforest, and I think most of us do, when you walk around the rainforest, you're going to see emergent trees, canopy trees, understory trees. We're going to see shrubs, herbs, epiphytes, parasites, and finally we're going to see lianas. And so in this part of the talk with Daniel Kisling from University of Amsterdam, we were interested to understand the evolutionary dynamics of a growth form such as lianas. Now lianas can be an ideal resting place for the tired botanist. Here my colleague Vincent Boissard taking a rest. A quarter of all plant species are climbers, 22%. Most of these are found in Calamoides subfamily, and most of them are found in subtribe Calamine, which now has become the megagenus Calamus. We've also heard a bit about this yesterday, that light for a little sapling at the bottom of the forest is a limited resource. So it's important for the plant to be able to grow quickly and to get to the top of the canopy to get enough energy to start the flower. I think we're at the ideal spot to see how tall the palm can grow. And I'm really looking forward Saturday to going and taking a similar picture as the one that Rodrigo took. Palms can grow to extreme heights, but they might be in a disadvantage when you compare them to woody plants. Here's a figure, and these little crosses here circled in purple represent palm species. And we can see that they don't quite match up to the height a woody tree, a woody plant can actually achieve. This is a logarithmic scale, so there's actually a huge difference here. We can see magnificent examples of sequoiadendron in California, which reach heights of about 100 meters. And this might be related to the fact that palms don't have secondary growth. And we saw again yesterday in Mayara's talk that this is actually limited during the life of the palm. But when palms evolve to lianas, they can achieve extreme heights. And they're actually the longest measured stem in the plant kingdom was a climbing palm, a rattan, measuring up to 180 meters. So this provides an incredible advantage to go straight up to the canopy and get the light they need to reproduce and thrive. These are supposed to be little circles. So here I represent a figure from our article where we showed the diversity of rattans throughout the tropics, and we can see that we have the highest diversity in Southeast Asia, mainly linked to the fact that Southeast Asia harbors a huge number of calamoid palms. And we can also see that canopy height, and this was a recent article which measured canopy height throughout the tropics, goes increasingly from South America, which has the lowest canopy height, to Africa, which is medium height, and to Southeast Asia, which has very high canopy heights, up to 80 meters, mainly related to the fact that the family Dipterocarpaceae, which is these huge emergent trees, dominate the landscape in certain parts of Southeast Asia. So in this article, we had several questions. The first one was, what are the environmental correlates of climbers throughout the tropics? And what are the evolutionary advantages? Can we find an evolutionary advantage of actually becoming a liana? And so for that, we used an integrated approach between macroecology and evolution. One of the first results that we got was that there's an inverted correlation between precipitation seasonality and palm species diversity. As rainfall varies throughout the year, we have a lower diversity in climbers. And this is interesting because it contrasts to what we know from woody climbers. Woody climbers have the opposite relationship. As rainfall figures vary throughout time, throughout the year, we have a higher diversity of climbers. This might represent the first evolutionary result that we had, is that there might be a phylogenetic constraint in palms. As I said, most palms belong to Calamoideae subfamily, and as I said at the beginning of my talk, most Calamoideae are restricted to tropical rainforests, to regions where there's very high rainfall and which is pretty constant through time. So most of these results is linked to the fact that most of the climbers originated in Calamoideae. We also found a strong effect on canopy height. As the canopy height increases, so does the climber richness. And this is something which was already obvious from the first slide, where we could see that canopy heights in Southeast Asia are extremely high. So the higher the canopy, the higher the diversity of climbers, which might be linked to the fact that they're able to get up there and able to get the light if they evolve to a climbing habitat. We were then interested in the evolutionary origins. So the first step was to understand how many times did the liana habits evolve in palms. And so using the dated supertree of palms, we inferred that lianas evolved at least 7 times independently. So there's nothing really new about this. The studies of Baker and collaborators in 2000 already suggested this. And so we had 5 independent evolutions at the generic level, and then 2 evolutionary trends towards lianas within the generic level, for example, in Dipsis and in Camadoria. So we were interested at this point to address 2 questions. First is, did the evolution towards liana to the climbing habitat have a general impact on diversification within palms? And did each time that we had an evolutionary trend towards climbers, did this have an impact for the diversification rates of that clade? So here I present an alternative view of the species level phylogeny of palms. This is based on the supertree, so relationships between genera come from the supertree. And then we just simulated random phylogenies using a Yule model for all of the genera up to the known generic diversity. This allowed us a quick and dirty way just to get a species level phylogeny. It doesn't really impact our results because we're looking at a trait that is constant or mainly constant throughout Genoa. The first result that we found is that yes, evolving to a climber habitat did have higher, they do have a higher speciation rate, diversification rate than non-climbers, and this to a ratio of 1.3. So here we have an estimation of the speciation rate for the liana growth form, and here we have the estimation of the speciation rate for the non-climbers, and this was a significant result. We then used another method, which is called BARM, and I introduced that a bit earlier, to look at the impact of the climbing habitat for specific clades that evolved that trait. And what we found is that we have an increase in Calamaini, which is represented here. So red colors represent higher extinction rates, and green colors represent lower diversification rates, and green and blue represents lower diversification rates. So we have a slight signal of an increase in diversification rates here in Calamaini, but none of the other climbers in Calamoidi or even in Desmoncus showed an increase in diversification rates. So this suggests that the evolution towards a climbing habitat does have an impact at the general level, but is not going to have an impact all the time at the clade-specific level. And this might be related to different morpho-anatomical characteristics of each of these clades. Lianas and the Calamaini clade do not have the same anatomical characteristics or morphological characteristics to climbing as those found in the African rattans, for example, ancestral line. I just want to take this opportunity to advertise a great postdoc opportunity, which was recently funded, recently granted to Daniel Kissling at the University of Amsterdam for 3 years, to use similar methods as those that I've shown here on lianas, but to look at frugivore evolution in palms. And I think there's a talk this afternoon, or there's a poster that's going to look at this in some way too. So it's a nice way to introduce this postdoc. So for those who are interested, please contact Daniel at his email, or you can ask Bill and I, who are also involved in the project, for more details. So to conclude, I'm not going to be too long on this. I hope that I was able to show you how, by having such a good data on the tropical rainforest family, it can be used as a good model to understand the origins of rainforests and the diversification patterns we find, both at species level and at morphological levels. This provided, at least from the last 3 years, a unique opportunity to understand the study of rainforest evolution and diversification. And interestingly, a lot of the data that a lot of the results that we have are starting to be confirmed in some way. So it's true that our analysis only represents one family, and but it seems like when we look at these meta-analysis, everything's going in the same way. In terms of perspectives, we already discussed this a bit at length at this conference. If we're looking at rainforest diversification, we have to look at more at species level phylogenies. I think we have a pretty good idea of what's happening at the global level, but we really have to have a better idea of what's happening at the species level. And we've had very interesting talks already, which are looking at this issue, what's happening at the species level, and I think we're going to have many more throughout this symposium. NGS data is going to be pivotal. We're really at a crossroad now where we either go ahead and produce a wonderful tool, which would be a species-level phylogeny of palms, which you can use to understand diversification patterns at an incredible depth. So one of the goals would really be to try and achieve this in some way or the other. And we're already discussing with numerous colleagues, and I back up what Bill said. We really have to work together as a community for this, which I think is what we're doing. And also suggest that looking at the dynamics of genetic diversity through time is also going to be an interesting pattern, because palms, with this very interesting intricate relationship with rainforest, might provide an interesting window to understand how climate change and future climate change will impact genetic diversity and species evolution in the more recent future. So I'd like to thank Rodrigo and Gloria for this wonderful symposium on palms here in beautiful Colombia. I appreciate their hard work. I congratulate also all the people who helped for this symposium. It's becoming a really interesting and fascinating symposium. I'd like to thank also all my co-authors that have helped me throughout these times. Thank you.